Hello, my name is Kellyn Sanders, and I am a Master's of Arts candidate in Medical and Biological Illustration. This is my presentation on my master's thesis, 3D Reconstruction of Fossilized Skull of South American Miocene Monkey, Homunculus Patagonicus, an Augmented Reality for Field Application. For most Miocene taxa, primate fossil evidence consists of broken cranial bones, teeth, and jaws. Studying these fossils is difficult due to the damage and distortion during geological stress. During fossilization, the soft tissue is seldom preserved. Homunculus patagonicus is an unusual primate from the Miocene epoch around 17 million years ago of extreme southern Argentina. To reconstruct the diet of such extinct mammals, the jaws, skull, and muscles of mastication provide insight into how food properties influence skull morphology over evolutionary time. The image on the right shows where the extant or living range of primates are in South America in comparison to where the Homunculus patagonicus fossils are being found in orange. One of the problems with bringing extinct animals to life for teaching, research, and public understanding is the incompleteness and distortion that characterizes nearly every fossil specimen. Even the best specimens are fragments, deformed, and/or missing parts due to the ravages of the fossilization process. Current teaching materials rely on simplistic line diagrams and photographs, which do not allow for detail of bony landmarks and delicate muscle fiber orientations. Digital resources such as three-dimensional, 3D anatomical learning applications are, at present, limited to human and canine anatomy. A 3D application for teaching extinct primate anatomy has never been attempted. This project allows the use of comparative anatomy to learn how Homunculus patagonicus lived and its relationship to its environment through an examination of dietary adaptations. The goals of my thesis were to complete a digital 3D model of the reconstructed cranium and mandible from the CT data, digital 3D jaw adductor muscle models, including origins and insertions based on extant analogs, a fully interactive iOS application, and finally a reproducible workflow documenting steps from CT data acquisition, digital reconstruction, and creation of the application. The primary audience for this application is scientific colleagues, PhD, graduate, and undergraduate students conducting research on primatology and mammalian evolutionary biology. The secondary audience is a broader interested public, and the tertiary audience is clinicians for use in comparative approaches for pre-surgical planning. For the materials and methods, we will go through data acquisition and processing, the cranium reconstruction, muscle reconstruction, origins and insertions, and the 3D iOS application development. During the data acquisition and processing, a micro-CT scan of Homunculus patagonicus fossil was acquired and segmented using Dragonfly ORS. Sediment remained after segmentation inside the cranium and needed to be manually removed using Cinema 4D. The turntable on the right displays how much sediment remained inside of the skull. During the reconstruction process, reduction of the distortion in the cranium was accomplished by referencing other specimens of the same species as well as extant models. For the first time, we can observe the skull of this species without the bias of distortion. During the muscle reconstruction, chewing muscles were completed after the cranium reconstruction, using the visible muscle scars on the skull and mandible. Muscle volumes were estimated using equations from extant South American monkeys. The chart to the right shows the muscle volume estimates. Muscle scars on bony surfaces of the skull and mandible were used to map the origins and insertions of each muscle. A in the image on the right shows the temporalis muscle origin in orange and the insertion in teal. B shows the masseter and C shows the medial pterygoid. Wireframes were created in Adobe Illustrator and then built in Unity as a user interface. Each section was designed and coded for while reusing the Illustrator design elements as sprites. My thesis resulted in CT data fossil model, reconstructed fossil model, muscle reconstruction models, extant distribution map, a phylogenetic tree, an iOS application built in Unity, and an animation demo. The digital fossil reconstruction of Homunculus patagonicus occurred in multiple stages, which allowed the specimen to be viewed for the first time as an average composite member of the species. We will go through the stages in detail. In the first stage, the manual sediment removal, followed by the reorientation and alignment of fragments, occurred to make the specimen as anatomically correct as possible. The second stage resulted in this digital restoration that filled in the missing anatomy and fragments. The reconstructed anatomy can be seen in dark brown and the original fossil CT data in light brown. The third stage resulted in a fully reconstructed digital model of Homunculus Patagonicus with as many data preserved as possible to showcase details of the bony landmarks and muscle attachment sites. The volumetric data were then used to create digital muscle models that attach to the cranium and the mandible of the reconstructed fossil. The muscles were constructed using their origin and insertion sites. The origin and insertion for each muscle group are seen here. The orange represents the muscle origins, and the teal represents the muscle insertions. The fully interactive iOS application was created featuring the 3D reconstructed models with augmented reality, original CT data, distribution maps of extant primates including the phylogenetic tree and fossil distribution map, and a placeholder animation. Now we will see the demo of my application and its different sections. The goal of this project was to reconstruct, for the first time, the missing anatomy of a complete fossil specimen of Homunculus Patagonicus. This reconstruction will provide a publicly accessible resource for researchers, students, and the interested public to view and learn about the specimen. The iOS application will be available from the Apple App Store and will allow access to everything seen in the demo. During the project, one of the challenges was the manual removal of sediment. The CT data provided were so impacted with sediment that the osteological details were impossible to visualize. Sediment inside of the skull created millions of polygons that did not allow the functions needed in ZBrush to complete the reconstruction. For this reason, it was necessary to spend a considerable amount of time removing the sediment manually in Cinema 4D by selecting groups of sediment individually and deleting them. A second challenge was the muscle reconstruction. Originally, the medial pterygoid volume estimate was 0.6 cubic centimeters. When this volume was used, the muscle was so small that it could not be extended between its origin and insertion. With this finding, Dr. Perry used comparative measures to recalculate a volume that would allow for a more proportional volumetric relationship to the temporalis group and masseter group. This new estimate was 1.3 cubic centimeters, which compared favorably to the data in Slade 2018. While more information is still needed to infer a specific diet, the reconstruction did provide visual verification that the dimensional data were realistic for the temporalis and masseter groups. For future inferences to be made on the diet, the specimen's heavy tooth wear will be taken into consideration. I would like to thank my preceptor, Dr. Perry, my faculty advisor, Jennifer Fairman, Sarah Poynton for helping during the Vesalius Trust and Thesis presentations, Kyle Erickson for helping get my app working correctly, Vesalius Trust for awarding me a Vesalian Scholarship, and my Art as Applied to Medicine faculty, staff, and students.